Hi, everyone. My name's Sebastian. I'm a degreeless heathen working out of a home lab in Huntington, New York. And I just started a kind of an oral history on what it took to get a PhD across the ages. So we're looking at folks dating back to as far as we can to people who have gotten their thesis today. And the overarching goal is to figure out what it took to do what they've done, a little bit more about what they've done, and some advice given their wisdom. So our inaugural episode is starting here with Wayne. So if you could please introduce yourself and let us know a little bit about your background leading up to where you are today.
Oh, sure. My name's Wayne Riekhof. I'm an associate professor at the University of Nebraska-Lincoln in the School of Biological Sciences. And I'm also courtesy faculty in the biochemistry department. And basically all that means is I can take graduate students through either program. And I'm also affiliated with a couple of other programs like the Fred and Pamela Buffett Cancer Center at UNMC. So I'm on the faculty there as an adjunct, I guess. And then before that, so like deep history, I grew up on a farm in Missouri, um, and went to the university. And I was always interested in science as a kid. I was an only child growing up, like out in the middle of nowhere, and my parents were a little bit older when they had me, and so I was close to my grandparents, my parents. But there weren't a whole lot of other kids around, and I don't know what it is. I just always kind of gravitated towards science, and growing up on the farm, you kind of have an opportunity to see it in all the raw detail as deep as you want. And I just enjoyed it. Um, and so when I was in high school, I kind of was into math and physics, and I thought maybe I would do physics or engineering. And then I did an engineering camp at the University of Missouri-Rolla, which is now called the University of Missouri of Science and Technology, which is like the engineering school of the Mizzou system. And I mean, it was fine, but I'm okay at math, but it's not, at the time at least, it wasn't like my niche. And so then I went to the University of Missouri. I graduated high school and from Odessa, Missouri, and went to Mizzou. And I started as a microbiology major. And the College of Arts and Sciences there and at Nebraska, it's kind of a theme for Midwestern land grant universities that if you're in the College of Arts and Sciences, there's a foreign language requirement. So I had to take a foreign language. So I took a 5-hour French class, and I got a D in my 5-hour French class. So that kind of tanked my GPA that first semester. And my academic advisor at the time looked at me in our first. We had faculty as advisors, and this guy at the end of my first semester kind of pulls down his glasses and looks at me and says, are you trying? Like, do you want to be here? And I said, yeah, I do. I really want to be here. And he said, well, biochemistry doesn't have a foreign language requirement because they're in the ag school. And, if you don't want to do French, maybe you can switch majors. So I did. And fun fact about that, the guy who told me to switch majors was George Smith, who won the Nobel Prize in chemistry in 2018.
Nice.
And this was decades before, well, this was at least 20 years before that happened. Um, but he was already pretty well known in the field. So he invented phage display. Um, and so anyway, so I got dressed down by a future Nobel Prize winner in my first semester as a very green freshman, fresh off the farm. But then I got into a research lab in the biochemistry department, and found out that was kind of the first time academically that I really thought I found what I want to do, because I was always kind of a mediocre student, but I could do kind of like a lot of us. Um, I did well in things that I liked, but I just kind of blew off stuff that I didn't like, and it kind of caught up with me. So I had to figure out what my niche was, and I had some mentors in the biochemistry department. One of which was a guy named Doug Randall, who ran the interdisciplinary plant group at Mizzou. He was a graduate of he got his PhD at Michigan State back in the mid '60s, I think. And so he, I was starting to think about graduate school and where to go. And I had done an internship at Monsanto in St. Louis, which I think anyone listening to this probably knows about Monsanto. And that was an interesting experience, but it kind of showed me that, maybe industry's kind of cutthroat. And the first experience as a Monsanto intern was showing up at a lab meeting on the first day and half the group being fired.
Oh,
um,
jeez.
or just reassigned. I can talk more about that. That's kind of parallel to the dissertation story. But so I went, so Mizzou, graduated, went to Michigan State, and I was really interested in plant-insect interactions. So I
Mm,
have a deep history. So growing up on the farm, I was involved in 4-H, which is like an ag kind of run by the USDA, and originated in the Depression as like a means for farm kids to develop marketable skills. But entomology was my main 4-H project, and so I kind of carried that. I was an entomology minor at Mizzou, and there was a guy named Greg Howe at Michigan State who was working on plant insect interactions and volicidin, which is when you have like tomato hornworms, for example, when they're feeding on a tobacco or a tomato plant. There's an amino acid, fatty acid conjugate that gets formed in the saliva that the plant perceives, and then they release methyl salicylate, which recruits parasitoids. And so you see those um, the tomato hornworms with like the pupae of the parasitoid wasps on its back. That's kind of the stuff I was interested in, like sort of signaling and plant insect ecology and chemical ecology. So I went to I applied a bunch of different places and wound up settling on Michigan State, and did a rotation with Greg in the summer of 2000 and enjoyed it. And I thought, well, that's where I'm going to wind up as a PhD student. And so then for my second rotation, so that was the summer. So then in the fall semester, I rotated with a guy named Christoph Benning, who was then um, an assistant professor. He had done his PhD at Michigan State from like 86 to 92. And then he went back and was a group leader at a research institute in Berlin for a few years. And then Michigan State recruited him back. And so I think I was his first or second graduate student after he came back to Michigan, like to join the lab. He brought some of his German students with him, and and so Christoph was working, had some really nice papers out on lipid metabolism in the chloroplast. This was
mm,
specifically the thylakoid, and I had been encouraged by Doug, the guy who told me about Michigan State in the first place, that Christoph was kind of an up and coming new assistant professor, but it was on lipids, and like taking biochemistry as an undergrad, I I guess I just kind of got the perception that lipid metabolism was solved and
mm.
everybody knew everything there was interesting to know about it. And so I did a rotation with Christoph anyway, and I kind of got hooked because it turns out that there was and still is a lot that's just not fully understood about membrane biogenesis and lipid compositions and how they're arrived at. So that was my second rotation. Then I went off and did a third rotation with a guy named Lee McIntosh, who sadly passed away with leukemia at the end of my PhD. But Lee was an interesting guy because he was he sequenced the first piece of plant DNA ever to be sequenced, which was the
Oh,
gene encoding one of the plastid encoded subunits of photosystem 2. And so he was a postdoc at Harvard in the early 80s and learned DNA sequencing from Wally Gilbert, who shared the Nobel Prize with Sanger for inventing DNA sequencing. I think that was like 1980. So anyway, so those are my rotations. And I just decided that I liked the Benning lab enough and the rotation project had gone well enough that that's where I landed. And so that sets the story for like me starting like the rotation project that I started on. I can get into that because that kind of formed the basis for much of the rest of my thesis or my dissertation research. But is that does that I don't want to just ramble on. So what
I,
do
I
you have any
guess,
questions about that so far,
yeah,
or
yeah.
comments or?
Well, no, that really sets the stage. What I'm interested in now is how you got to the project you finally chose. Like, what were the offshoots that you tried, if that was the case, and more importantly, what was your decision process like? Like, how did you, or was it just serendipity, things like that?
Well, and so this, for folks who might be watching this, trying to decide where to do a PhD or who with or what to work on, it varies a lot from place to place, and especially it matters how you're funded. So when I came in, I had done a lot of undergraduate research, and I had I had already had a publication, and which is more common now, but it was fairly uncommon at the time, I guess. Um, and
What year
so
was
I got
this?
an.
When?
This would have been. I applied in the fall of '99 and interviewed in the winter of 2000. And so, just in terms of that process, I applied to let's see, Cornell, Wisconsin, Michigan State, Penn State, Washington State, and Mizzou. And I got in everywhere, actually, which is surprising for someone with I wound up with like a 3.4 GPA, which is, it's not terrible, but nothing to write home about. But I think people could just sense that I was really into research and I was pretty good at it. And that, to me, as someone who recruits graduate students now, that's what I look for. I don't really look at GRE scores or even GPA. For the most part, I kind of just look for that spark of like someone who's really interested in what they're doing. And, they've shown me that they actually are interested in what I do. And, and sometimes, rotation students will come through the lab, like the student I have that's finishing now was a rotation student who didn't really have a whole lot of interest, but we kind of hit it off during her rotation, and she's done a great job, and we're starting to publish all of her papers now. Um, and so, in terms of after I made the decision to go to Michigan State and I did my rotation, settled in the Benning lab, I had gotten a plant research lab. So at Michigan State, and this is still the case, Christoph Benning's he's now the director and has been for a few years, of the Michigan State DOE Plant Research Lab. And so that was founded in 1965 by what was then the Atomic Energy Commission, which eventually became the DOE. And the goal was, like, this is the height of the Cold War, the goal was to understand what happens to plants with like nuclear fallout. If there was a nuclear war, how,
Cool.
what happens with plants? And and so it's been continuously funded for almost 60 years now. And so I got one of these nice fellowships that paid basically the PI, whoever I wound up with, didn't have to pay me because I had my own money from the DOE fellowship, and then I got another fellowship from the state of Michigan. And so that mattered a lot because it let me do whatever the hell I wanted. And Christoph really encouraged that. And so the rotation project I took on was one that had been initiated by one of his German master's students. And so the idea was, and this is something that the Benning Lab had been working on for a while, because Christoph kind of rediscovered this phenomenon during his PhD, that phospholipids are sort of the canonical thing that people think of as being what biological membranes are made of. It turns out that plants make the most abundant membrane lipid on Earth is a non-phospholipid. It's a glycolipid with a galactose as the head group instead of a phospho base or something.
That's weird.
And well, it makes sense because phosphorus in most, especially in aquatic environments, is oftentimes the limiting nutrient. And so efficient phosphate use efficiency is one of those things which has just been under hard selection for the last 4 billion years. And so it turns out that when you take many bacteria, especially soil bacteria, fungi, algae, seed plants to an extent, if you put them under a phosphate-starved diet, then they turn over phospholipids and they make a bunch of lipids and different kinds of lipids that don't contain phosphorus. And so that's what I started working on, using the purple photosynthetic bacterium Rhodobacter spheroides as a model system. And I think there was I think the BlueSky thread that may have prompted part of this was you talking about the mess that it is working with high GC organisms and getting PCRs to work and stuff like that. That's something I had experienced as an undergrad, because I did undergraduate research with a guy working on corn, and just PCR. I mean, for whatever reason, maize is incredibly high GC in specific regions of the genome, and so I had gotten used to working with high GC DNA and cloning and PCR and stuff like that. And I think that was one of the things that impressed Christoph right off, was because in my rotation, I knew how to design PCR primers. I knew how to cope with an organism that has like 75% GC, and everything just worked. And
Wow.
so when I said, would you mind if I joined your lab? He's like, yes, do it. Let's do it. And so I picked up that project and just kind of ran with it. And then the goal was, and of course, your moniker of a tiny green cell. I was working on a tiny green cell called Chlamydomonas.
Sure.
And so clammy is,
Legendary green cell.
yeah, right. Um, and so Chlamydomonas is basically, well, it's one of the few eukaryotes which does not make phosphatidylcholine. So phosphatidylcholine is sort of the canonical membrane lipid that people think of when you think of membrane lipids. And clammy doesn't make it at all. It makes phosphatidylethanolalamine, which is like PC without the N-methyl groups. But um, it makes this other lipid, so this betaine lipid, which is diacylglycerol trimethylhomoserine, which is basically a chemical mimic of phosphatidylcholine, but it doesn't have phosphorus. And so the goal of the Rhodobacter work was to identify the genes, and that's let me take a step back. When you take Rhodobacter and starve it for phosphate, it switches from making phospholipids to making glycolipids and this betaine lipid.
Hmm.
And so we identified the genes, and part of one of the papers in my thesis, I think it was like chapter 3 maybe, was doing the biochemical characterization of those enzymes, expressing them in E. coli, partially purifying them, developing an enzyme assay, which was all, sort of core biochemical stuff that people still do, but that was just a little more common in terms of like direct uh, development of new assays and things like that. So I did that. And then it was like early 2003, Chlamydomonas was the first algae to have its genome sequenced. And so I got early access to the genome sequence. And then there was a consortium of folks associated with the Joint Genome Institute of the DOE in Walnut Creek, California. So that's where like basically all of the DOE's genome sequencing center is based. And so there's a huge number of genomes that they've sequenced now, but Clammy was the first. And so I got in on the ground floor of that because Christoph was asked, Christoph Benning, my advisor, was asked to go to Walnut Creek and attend the Genome Jamboree, which is what DOE, the Joint Genome Institute, used to do. Was just bring in everyone who was working on a particular model organism, and you get early access to the genome. You sit and annotate all of your favored pathways, but Christoph didn't want to go, so he sent me, and so that was sort of a foundational thing for me, like getting networked into the clammy community. And there was a paper in Science a few years later. So Sabia Merchant is at UCLA still, and was the lead on that, and Arthur Grossman at Stanford, and and so that came out a few years later. But having that early access, I took the genes that we had identified in Rhodobacter and just did a blast search of the clammy genome, and I found homologs. But the interesting thing was that, and this is a common theme that I teach in my classes, just because you see it over and over again when you look, is that there's lots of prokaryotic biochemical pathways where you have an operon that is a polycistronic transcript that might encode 10 different proteins that all function in the same pathway. And so the betaine lipid pathway, there's two genes encoding two proteins with 4 biochemical functions, because the methyltransferase can do all 3 methylations. I mean, if you just look at the pathway, you'll see what I'm talking about. But and so in bacteria, there are 2 open reading frames on the same transcript. In eukaryotes, they've been fused onto the same protein. So it's a single gene that encodes a protein with 2 domains, and that basically does the whole pathway.
Is there like a
And
linker in
so,
between or something to keep the
yeah, there's
domains?
kind of a flexible linker, and I tried to purify it and start to try crystallization trials and stuff during my PhD, but I just could never it's not an integral membrane protein, but it's enough of sort of it's got a couple of amphipathic helices that seem to be unstable when they're solubilized.
Hmm.
So I could never even purify it to get to the
Wow.
point of trying to crystallize it. But now that AlphaFold is out, the structures are pretty much identical across, and there's examples of this betaine lipid synthase um, from fungi, a bunch of different algae, mosses, and they're all basically superimposable. So I'm pretty confident about the structure. But yeah, there's a flexible linker between the two domains, and but then I kind of genetically separated them. So I expressed the domains separately in E. coli to show that their function is the same as they are in bacteria. So that was my second first author paper, I guess. Um, and then like along the way, one of the ways, so I'll take a step back. So first of all, I should stop because I like to talk about this stuff, and
No, no, this
and
is great.
you
This is great.
I'm sure I won't be the first to, when you ask someone to talk about their PhD research, it's like the ego says, "Oh yeah, you want to hear about that?" So so stop me. Feel free to stop me
Sure.
if I'm going too far off base. Um, so let's take a step back. So I characterized the genes in bacteria as a bridge into eukaryotes. Before that, we had developed um, a biochemical screening technique where we had a robot that would spot thin layer chromatography plates. So we were doing basically shotgun gene tagging in clammy. So just transforming with a nitrate reductase. So the wild type strain of clammy can't use nitrate as a nitrogen source. And the reason is because it's mutated in the nitrate reductase. That's the entry point for nitrate assimilation. And so we had a plasmid from a guy named Don Weeks who just retired from the University of Nebraska. So he was my colleague, but before that, we had gotten plasmids from Don, years before I knew I was going to be on the faculty here. Uh, and so I developed this gene tagging approach, and there was a paper from I think like 2003 in Plant Physiology where we described that technique, and I didn't find any mutants that were defective in the synthesis of the betaine lipid. It turns out that's an essential gene. But what I did find was a sulfonic acid containing lipid called sulfoquinovosyl diacylglycerol. So that's another thylakoid membrane constituent that that is inversely related to phosphate starvation. So when they have enough phosphate, they make less sulfolipid. When when the algae or plants run out of phosphate, they stop making phosphatidylglycerol, which is the other anionic component of the thylakoid membrane lipids, and they make this sulfur-containing lipid. And so we identified a gene that is involved in the pathway, and that was actually known from Arabidopsis. But the interesting thing was, in that paper, I learned how to do NMR on glycolipids, which is actually it's kind of like doing high GC PCR. It's one of those things where it's not quite as easy as doing just sort of like normal glycoconjugate structures. Like glycolipids are hard, so I learned how to do NMR on hard to NMR molecules. But I got that to work, and there was a variant of the sulfolipid that had an extra fatty acid esterified on one of the sugar ring hydroxyl groups, which was new, and we still don't. I mean, that's over 20 years ago, and we still don't know why they make that. But it turns out that most plants make that variant. It's just not clear exactly what the biochemical pathway is that puts that second fatty acid or that third fatty acid on there, and what the function is. But so that was a paper. That was another paper for my thesis that sort of tied together the way I still like to do biochemical genetics, where and you're going to have to edit out some of the ums because I'm thinking on the fly here. But
No, you're good. You're good.
um, um, so so Christoph Benning's approach was similar to that of his PhD advisor, who's a guy named Chris Somerville, who, along with Elliot Meyerowitz at Caltech back in the 80s, they basically developed and popularized Arabidopsis as a model system. And and so, for most of the 90s and early 2000s, most of the folks who were working in Arabidopsis were trained in the lineage of those two labs, either Meyerowitz or
Hmm.
Somerville. And um, and so what Somerville did was to basically say, well, we've done all this biochemical genetics in fungi, like going back to Beadle and Tatum and the one gene, one enzyme hypothesis and all that kind of stuff that you learn about in a genetics class. Somerville took that approach and applied it to plants, which hadn't really been done systematically until around the early 80s when he started doing that. And then that all of his students basically took those tools that were developed in Arabidopsis and applied them to pathways of interest, hormone signaling, lipid biosynthesis, cell wall biosynthesis, et cetera, et cetera. And so that's kind of the intellectual framework that I grew up in as a graduate student, was learning those approaches that we're going to do biochemistry, but in order to identify the interesting biochemical pathways upon which to work, we can use genetics to identify mutants that have defects in those pathways, use that as an entry point to identify the genes, and then go from there. And and so that's basically what I still do, just in a more systematic way in Saccharomyces, because instead of needing to do classical forward genetics, we have knockout mutant collections where I've got in my lab right now. We have basically all of the non-essential genes in yeast. We have mutants. So we've got like
All of them.
4,800 different, yeah, all of them.
Jeez.
Yeah. So we've got like, I think it's like 50 96 well plates. Each well has a different mutant with a different gene that's knocked out. And so it makes, biochemical phenotyping like super simple. And we're kind of developing that for undergraduate teaching labs to as a bridge into as sort of a cure format, so a course-based undergraduate research
Yeah,
experience
I was
type
about to
thing.
say, I'm like, that's perfect cure material to just like run
Well,
through that. Yeah.
if you look up recent papers from my lab, there's a lot of there's several from undergrads as first authors on
Hmm.
using those tools to one of the things we're doing now is trying to understand uh, well, basically a class of chemotherapy drugs that disrupt phosphatidylcholine biosynthesis, and then looking for strains that are either resistant or hypersensitive to those compounds, and asking the question, well, what genes become essential when we challenge the
Hmm.
cell with this phospholipid
That's cool.
biosynthetic disruptor? But all of that, I can trace everything back to, like that approach. I trace all that back to my first couple of years of grad school and learning those tools, um, which again kind of go back to the 1930s when George Beadle and Tatum were working on this at Caltech. And the funny thing is, the the building I work in, that my lab's in at UNL, is the Beadle Center,
Hmm.
because George Beadle is a native of Nebraska. Went to the University of Nebraska as an undergrad, and then went to Cornell and was a contemporary of Barbara McClintock and a couple of other people. And there's a famous picture of uh, the Maze Genetics group at Cornell in like 1925, and there's like out of five people, just random graduate students in the field, there's like three future Nobel Prize winners in that one picture. And so it's kind of fun because the work we're doing now, which kind of some of which follows directly from what I was working on the non-phospholipids in algae. Fungi also make those. So Neurospora crassa, which is the organism that Beadle and Tatum worked on, they actually identified some phosphatidylcholine pathway defective mutants in Neurospora. So when I started my lab, I ordered a bunch of Neurospora mutants, some of which had been deposited by George Beadle in the 40s. And so I just thought that was kind of cool. I'm here
Wow.
in, it would have been like 2014, 2013 at the time, um, in the Beadle Center, named after George Beadle, working on Neurospora mutants that Beadle himself identified and deposited in the Genetic Stock Center. And there was a paper from, I think, 2014 I didn't wind up using any of Beetle's strains because I just sort of got the clean knockout versions of those strains. But still, it's kind of
Yeah, there's
I
like a
just I
physical
like that.
lineage. Yeah.
Yeah, no, exactly. And that's what, when I teach. So I teach two classes. I teach a molecular biology class that I'm kind of merging a lot of cell biology into, and then I teach a graduate level microbial physiology class. And so I like to pepper in the history of all of this stuff. And I think some students are sort of like, well, who cares? But there's some people who, I really kind of are interested, because one of the classrooms that I teach in is one of the first big lecture halls that was built at Nebraska. And it's almost certain that Beetle would have taken classes in that building.
Wow.
So when I'm teaching, Beetle's work and how it relates to what we do now, and I say, and one of you might be sitting in the same space that Beetle occupied 100 years ago, I mean, it kind of just gives you that direct connection in a way that you wouldn't get other places.
Yeah, I
Or
remember
or
my background being very like every time I would take a class, there's always this kind of abstract thing, like, okay, now you're going to learn how the globby gloops interact with the bleepy blops, right? And
yeah,
I'm just like, yeah, okay, I get it. But having that historical lens, at least from my very not conventional sense, that historical lens like ties me to the research in a way that's very different from just kind of learning for the sake of an exam, if that makes sense.
right,
Yeah, yeah, very very
right.
cool.
Yeah, and I try to steer, I'm getting into pedagogy now, but like I do try to steer away from that, because I feel like, well, if you're staying up until 2 a.m. to cram for an exam and you're going to spit everything out and forget it as soon as you leave the room, then what's the point,
Right.
So, but anyway, okay. So going back to the whole, like, the thesis part of this,
Yeah.
or the dissertation thing that you were originally asking me about. Um, so I did my PhD relatively fast, and that was partly because I had good experience as a technician. So I came in with good hands. I knew how to handle organisms. I knew how to I didn't ever have a formal microbiology class, but like I was pretty good at the bench. Still am when I get a chance to go back to the bench. And so I came in with good skills, and I was in a pretty good sized lab. I mean, Benning's lab at the time I joined was probably 10 people, and it got up to about 20 people by by the end of my time there. And so there was lots of senior students and postdocs to learn from, as well as the boss. And the plant research lab at the time was just, bustling. Like, there were two National Academy brewers, Hans Kende and Jan Zavart, who were plant hormone biochemists. And, but then, it was just a really fantastic place to train, because, like, the science was just really good, and everybody was pretty open and collegial and well funded. And so you want war stories? I gotta say, I don't have that many war stories. I think it's maybe part skill, but a lot of luck, just being in the right place at the right time. And you always refer to yourself as a degreeless heathen. I mean, that's kind of how I felt going into this, because I was just some random kid off the farm in Missouri who, like, I liked science, but I'm a first generation college student. I didn't have, my parents didn't. They kind of knew what I did, and my dad has since passed, but my mom's still living, and both my parents are smart, but they're not scientists, and they didn't have any means to sort of advise me on what to do. And I kind of remember, I don't know why I was thinking about this the other day, maybe because I was writing a couple of letters of recommendation. But when I was applying to graduate schools, I think I mentioned to my mom that people were writing me letters of recommendation. And she knew our congressman
Mm.
at the time, who was, not a close friend, but it's a fairly small rural community in Missouri, and so everybody kind of knows everybody else. And she says, "Well, do you want me to get Ike Skelton to write you a letter of recommendation?" And I said, "Well, I'm not going to West Point. I think that's what
Right.
you need, like, a congressman for." So I kind of just stumbled in to a lot of these opportunities, and um, I don't know how much of that is luck and how much of it's skill and how much of it's privilege. And
Yeah.
but
we don't really know. Yeah, there's no good way to really figure that
Yeah,
out, right? But like, I guess one of the most important questions regarding that type of background is for students today that are applying to PhD positions, and they may not have like the quote unquote good hands or the lab
right.
skill experience to be a technician prior to accelerate things. What
Sure.
would you recommend they do to either skill up or to yeah, just in general, like what kind of advice would you give looking back?
Well, I think it depends on what kind of institution you're at the moment. So I was at, Mizzou at the time was like sort of a mid-tier Midwestern R1 university, kind of like not quite to the level, I'd say on par with Nebraska, maybe a little bit better. Not quite to the level of like a Michigan or a Michigan State or an Ohio State. Um, but I certainly had the opportunity to do research in labs. And um, that's kind of the key thing. But a lot of people don't have that opportunity. And so if you're at a PUI, a primarily undergraduate institution, um, there are opportunities. So like NSF research experiences for undergraduates. So NSF programs. Um, Nebraska has had one for about 10 years now. It's on hiatus this next summer. But then one of the other faculty, so the guy who ran our REU on bioenergy systems, which is kind of the area that I was working in for a long time, has retired. And so we're kind of regrouping and turning it into an industrial microbiology research experience. And so there's lots of those types of opportunities. And if nothing else, it gives you, if you don't have the opportunity to do research in like an active research lab at your institution, then it gives you a bridge into seeing what real research looks like and the kinds of frustrations. Now, I should say, I said I don't really have any war stories. There are some. I mean, it's not a cakewalk and it is hard. Um, but that said, like I feel like, like I said, I was set up for success early on because of just sort of stumbling into the right place at the right time. And I think just being aware of what's out there
Hmm.
is important. I hear from people that, you might not realize that you get paid to go to graduate school, or you should. I mean, you shouldn't have to pony up your own money for tuition and living expenses because you'll either get paid from your advisor's research grant or you can teach lab classes typically. So my students in the School of Biological Sciences at Nebraska typically do a mix of both. And so usually I pay um, a research assistantship for one semester, and then the student teaches typically the microbiology lab courses, which are just right next door to my research lab for the other semester. And then we have to come up with summer funding. And so it's always a struggle to sort of monkey branch along funding to make sure that everybody gets paid. That's, like sort of in thinking about things that keep me up at night, that's number one, is just making sure I have the funds to
Yeah.
keep the lab supplied and keep everyone paid. Um, but you might not know that, or you might not know you might not know what you don't know until you sort of have someone sit you down and say, this is how a graduate degree in a biological sciences works. And so it's a good idea to try to do an REU or some similar type of experience. Um, there are NIH funded programs through the INBRE program. Many universities have sort of localized research experiences. So here at Nebraska, um, football, even though we haven't been good at football for a long time, the sports now volleyball were great, but um, Pepsi actually pays the university for a monopoly on soft drinks on campus and at sporting events. And to maintain that monopoly, they give us funding for undergraduate science programs.
Wow.
So, well,
Shout outs
and
to Pepsi.
across the yeah, no, yeah, no, it's the UNL UCARE program. So it's the undergraduate creative activities and research experience program. That's the acronym. And so that, that pays a small stipend and a little bit for research funds for 2 years. And for undergrads here, the program is actually pretty well funded. And so it's sort of like money that you just have to know is there so you can ask for it. And I don't think I've ever had a student, you have to write it like a one-page application, just sort of describing the project. But I don't think I've ever had anyone who didn't get it if they applied for it. And so there's a lot of programs like that that would be specific to your own university or um, opportunities that you might have to go digging for a little bit. So I had um, Jackie Garcia, who's on a first author on a paper from maybe 2021, I think. She's a PhD student at Emory University, no, at WashU in St. Louis, and she was just phenomenal at funding her own undergraduate research. I mean, she sought out opportunities and got them from places I'd never even heard of. And so, I think just showing that you have the initiative to go out and find those opportunities, and that's important. But I think if you don't even know that any of that exists, you've got to have someone, um, to tell you, and that's on faculty like me to make sure people are cognizant of that.
Right.
And so that's my take on it in terms of, yeah, I feel like I'm way off base of where, or maybe where you started, but
No, you're good.
um,
You're good.
that's um, that's my take on it.
I
Is
guess
that
a
if,
good.
if you're not in
Good.
if you're not in an R1 university, there are still opportunities out there. You just got to look a little harder for them, maybe.
Gotcha. And from to flip it to like you're mentioning faculty, to the professors and educators watching this, what advice would you give them to help better prepare students for potential research positions, especially if they work at PUIs?
Um, hmm. So I'll give you some folks who might be better at answering that than I am, just from my own network, and I can give you those after we're done recording, or I can
Sure,
just
sure.
give them a shout out here. So one is a guy named VJ Rubenstein at Ball State University in, I think, Muncie, Indiana. And so VJ and I have known each other for a while. I think we probably connected on Twitter, but we're kind of in the same. We kind of work on similar things at this point. So yeast membrane dynamics, and he works on various topics in vesicle transport and stuff. But Ball State is a PUI. Now they have a master's program, but um, I think it would probably you'd probably be better asking people who actually have that experience of directing undergrads at PUIs into research programs, or like your buddy in the Blue Soup stuff, Aline.
Yeah.
I think she's pretty good at that. And Terry McGlynn he's one of the CSU campuses. I forget. Um, but it's just basically what I was saying. Like making your students aware of what's out there is really important, and especially if you teach lab classes, um, sort of planting that bug that this is something you can do. Mark Martin at University of Puget Sound, who
Hmm.
I think is nearing retirement or maybe already retired, but he does a great job at this as well. Taking folks who are at a PUI who have a research interest and getting them that experience and making them aware of what's out there and what's possible. And I just it's not. I don't think it's the same type of impediment that it could have been 25 years ago when I was starting into this. Um, and I'm trying to think in terms of my grad school cohort. I've got friends who went to Purdue, UC Davis, Mizzou for myself, Wisconsin. So um, a lot of R1 state universities and things like that. And I don't. There were a few, I think, who went to smaller regional PUIs, but not as many. And in terms of the, we're going to be interviewing graduate students here in early February, and so we get a lot of applicants from maybe not the flagship university, but like smaller schools in Iowa, South Dakota, Missouri, Kansas. Um, and these are all folks who oftentimes have done an REU program at Nebraska. So that's one of our recruiting tools essentially, is to bring people onto campus for a summer and introduce them to the work that's going on here, and then try to recruit them back for graduate school because they already have experience with folks here. They've been to Lincoln, they know it's, you know, kind of a smaller Midwestern
Hmm.
small city, but it's a nice place to live. And but in terms of specific advice to faculty at PUIs, I think there's, just in what I've said, but then there's a lot of folks who you should reach out to. Just some of those names that I mentioned, and folks you and I know from Twitter who do a good job advising undergrads at PUIs.
Cool.
Yeah. So
Very, very cool. Thanks so much for that.
yeah,
Um,
yeah, you bet.
Now, I guess one last question would be if you could meet yourself in the past and give yourself one bit of advice that could have potentially changed something or given you a leg up. Either it could be technical, it could be purely academic. What would it have been if you can give
Huh.
yourself some advice?
I would say don't. This is going to sound. I don't know. I don't know if I want to say don't stress out so much, but because science has gotten way more competitive at this point. During the 90s, with the NIH doubling and with the expansion of NSF and DOE and those programs, you would think that expansion of funding would have made the competition for grant dollars less. It seems to have done the opposite, because the more people you train, the more people are in that pool competing for the next batch of funding. Um, and so I spent a long time really anxious and really mm, stressing out. Um, and I wish I hadn't, because looking back, and again, this is just my experience. Like, things weren't. I don't know. I stressed out about the wrong things, maybe. And you can. I'll be honest. Okay, you can edit this out if you want. I'm okay with you including it. I think I made a joke when you were talking about dissertation writing, that Jack Daniels and Jim Beam are really bad writing partners. Well, what wound up happening is I wound up with a drinking problem, and I had to manage that, and I had to, basically admit to myself that I had a problem and that I had to fix. And a lot of that I can trace back to my anxiety and stress over getting grants funded, getting papers published. And it's a thing that a lot of people fall into, surprisingly. And I'm trying to be open about my recovery and the fact that it exists.
Sure.
And, and I kind of make jokes about it now, but um, I've known people who've died, who've drank themselves to death, who,
Wow.
and it's not. I joke about it, but it's not a joking matter. And that kind of just the constant anxiety and worry, and I'm kind of a nervous temperament by nature, I suppose, which and I've also been diagnosed with ADHD, and it turns out that about 50% of people who have an ADHD diagnosis will develop a substance use disorder at some point in their lives. And that sort of self knowledge, I can look back now and see the mistakes that I made. But in all honesty, the honest answer to that question of what I would go back and advise myself to do: don't use alcohol as a means to cope with anxiety.
Wow.
And that's there's some sort of deep family history there that I didn't know about. And then I did an ancestry DNA test, and I got I downloaded the SNP data, and there's some third party um, tools to analyze it. And it turns out that there's like three SNPs in a cluster of genes on chromosome 12, which are all alcohol and aldehyde dehydrogenases. And there's three SNPs that are really, really, really predictive for alcohol use disorder, and I've got all three.
Wow.
And and my my grandparents' generation. So I'm from a Southern Baptist, very small religious community in Missouri. No one drank. I didn't know anyone growing up in my family who drank alcohol at all. And it turns out that that's because all of my great aunts and uncles saw my great grandfather basically drink himself to death. And he was just a nasty, abusive drunk. And that's one of those things that the next generation saw that. Everybody sort of said, I don't want anything to do with this, but I didn't know that about my family or about my genetics. And, I mean, genetics loads the gun, environment pulls the trigger. So I don't mean to get off on a recovery thing here, but
No, this is important.
it's important. And that's why, um, I think substance use disorders like that are less stigmatized than they used to be, but there's still sort of a guilt or at least on my part, like it's like, well, why did I let myself go down that road? It's just sort of a very gradual thing. You don't quite realize it while it's happening until it's kind of at the point where it's like, oh, well, yeah, I got to do something about this. Um, and so, that's another thing is just even though I had a good experience in graduate school and as a postdoc and, mostly as a junior faculty, um, just the stress of the job and the grind and keeping things moving forward, um, developing effective coping strategies is important. And, at some point, I don't know what happened in my own brain, but, something flipped. Like there was a tipping point where um, that happened, and I'm better now, and it's under control. But uh, just being cognizant of those types of issues. Like I used to run marathons. I used to do long distance rides on the bike when I lived in Colorado. Um, but I can just see now that I've had to kind of go back and look at it. I always had sort of an unhealthy relationship with alcohol, especially, and it's just important to be aware of that. And I don't see that as much with graduate students nowadays, um, but weed is legal almost everywhere, and I see a lot of not necessarily problems, but I kind of hear things, and that I think is not as damaging physically as alcohol or other drugs, but it still can be, sort of an unhealthy way to deal with stress. So, like, I don't think I would do anything differently academically or like in choosing the things that I work on or the experimental approaches, that kind of thing. It's just having self knowledge to know how to cope
Mhm.
with that kind of stuff.
Wow.
And and so I've hesitated to be super open about that, but there it is. Like
I appreciate that so
I
much.
yeah,
I'm sure that's going to resonate with a lot of people, especially because
sure.
from all the stories that I've heard, the stress, how one copes, and how that monster of stress impacted the entire research trajectory of people in grad school, it was a significant topic, and not a lot of people talk about it. So, like,
Yeah,
seriously, thank you for the vulnerability. Yeah.
yeah, well, that's I when I realized that I needed to, obviously stop drinking and like sort of work on myself to figure out how to cope differently. Um, my lab members knew that things were different, or that I was, I was still teaching my classes, I was still writing papers, I was still getting grants in, but I just wasn't firing on all cylinders, and so I just sat the folks who I needed to tell down, and I said, "Here's what's happening." And all of my colleagues, all of the people in my lab, were very supportive and have been along the way. And so that's it is. I mean, thank you for acknowledging it is a vulnerable thing to admit that, but I'm trying to use my example so maybe someone else hears that, recognizes something in themselves, and says, okay, that's all I have to say about that. But that's definitely one thing that I would have coached myself through is, and I think I just didn't know that I had that tendency, or I kind of saw it as normal.
Mm.
Because especially conferences, especially Gordon conferences that I used to go to, and that I still go to sometimes. It's just a normal thing. The conference goes until 9 o'clock, and then you go to the bar and have some beers, and everybody loosens up, and you talk science. And I've had a number of good collaborations who came about from conversations at bars, at conferences, and that's great. Um, it's great if you are not an alcoholic and don't have that tendency, but it's just something to watch out for.
Right.
So,
Wow.
yep,
That's huge advice. Thank you again. That was
yeah,
awesome.
okay.
On the closing note, is there anything you'd like to plug or acknowledge, or an anecdote? Anything you'd like? The floor is yours for the last couple minutes.
Well, so I was just thinking about this because I sent you the PDF which is from the Michigan State Libraries, and I hadn't looked at it for, well, I mean, I hadn't looked at the acknowledgments, and so the acknowledgments of my dissertation. So I worked with two really talented undergrads, one of whom, Todd Leidick, is he's the director of the mass spec core, or one of the mass spec cores at Michigan State, and another guy named Mike Ruckel, who was on the faculty at ETH Zurich. So Mike is from rural Michigan, but then he wound up doing a postdoc in Switzerland, um, and staying on, and now he's in industry. But two really phenomenal guys who I were undergrads with me as a graduate student. Um, the story. So there's a story that we still tell, where so we were doing a chlamydomonas genetic screen, and the mutant that we found, that we wound up publishing on the sulfolipid biosynthetic mutation, or the SQD1 mutant, which is from Plant Phys in 2003. Um, so one of the things you have to do when you do that type of screening is to make replica plates. So you make your mutants, you plate them out, you get single colonies on plates. Each colony is going to have a plasmid insertion into the genome at a different locus. And so what we did was to take toothpicks and pick those into deep well 96 well plates. And so like 2.5 mils. So you get these little mini cultures. And then we had a 96 prong replica printing device.
Mhm.
So we would just sample each well in parallel and make a 96 well print onto an agar plate. And one of the guys was reading TLC results over the weekend and found a mutant that lacked that lipid. So that was interesting, that we knew that was something that we would probably want to characterize further. And we came in the following Monday and looked for the replica plate, which we had this sort of lighted shelf that all of our algae plates were incubated on, because I was the only one working on algae at the time. And the plate was gone. And it turns out someone had thrown all of the replica plates mistakenly into the autoclave bin.
Oh.
And we had the custodial service at the time at Michigan State actually handled that for us. So they would take the bags of petri dishes to the autoclave, and that usually happened over the weekend. Well, luckily that weekend, the custodial staff didn't empty the autoclave waste. And so we dug through the trash to find the replica plate. So we called that TCM1, which is the trash can mutant. And
Nice.
that wound up, and we wound up, that was my first author publication
Wow.
as a graduate student. And, so it's like little serendipitous things like that, where someone makes a minor mistake, or there's a misunderstanding about which plates we need to throw, which we need to keep. And then just by a quirk of fate, the custodian who would have normally done that was sick that weekend and didn't do it. And otherwise, I probably wouldn't have graduated in 4 years.
Wow.
So,
Wow. Wow.
yeah,
Wow.
it's funny how those kind of historical contingencies happen. And you just recognize that again and again once you see it, and once you know to look for it. Uh, and, I don't know, I like things like that. And when I started my research lab at Nebraska, my first REU student um, was a girl named Ashley. She was at Fort Valley State University in Georgia, which is an HBCU. So it's her first time doing research, and she did a great job. She came to me one day. We were doing a screen for fatty acid defective mutants in Chlamydomonas. And I had just bought a gas chromatography setup. We were calibrating the thing. I had a whole series of wild type strains of clammy that we were using to just sort of calibrate the machine and make sure we could separate all the fatty acids we wanted to screen for. And she came to me, and there are 4 different omega-3 fatty acids. So fatty acids that have a double bond, 1, 2, 3 carbons from the end. And it was missing those 4 peaks. And so she comes to me and says, I think there's something wrong with the machine, or there's something wrong, because there's like these 4 peaks aren't there. And I looked at that and I said, do it again. And so she ran the sample again. She made fresh extracts and ran the fatty acid methyl esters again. It was the same thing. And it turns out that through a weird quirk of who knows what type of evolutionary selection, there's a single omega-3 desaturase in clammy. So higher plants have like 4 of them, and in different compartments. Clammy, it turns out, has just 1. So that was the first paper from me as an independent PI, was on an absolutely accidental discovery of a wild type strain of clammy that just so happens. So clammy is one of those organisms that people don't normally freeze it. So you just sort of serially passage it for 50 years.
Sure.
And you get basically a bunch of different laboratories that all have their own isolates of all of the different wild type strains that have been serially passaged in parallel for decades. And, it turns out there's a lot of interesting sort of mutational profiling you can do from those strains that people have done. And we just happened to discover that one of those wild types had a mutation in the sole omega-3 desaturase of clammy. And so that paper came out in Eukaryotic Cell in 2014, I think. And that's since Eukaryotic Cell has since been rolled into MSphere. It's an ASM journal. But that's just, it's just sort of accidents. And I tell those stories in class because I also cover like the discovery of penicillin by Fleming, which was pure accidental. But, it's one of those things where I think Pasteur said chance favors the prepared mind, right? So if you just sort of take a step back, and when you see something odd, or if you just, maybe don't throw all your plates away right away and let them sit around. My former colleague, Steve Harris, who's now chair of microbiology at Iowa State, used to refer to this as benign neglect. You just sort of let stuff sit around.
Oh, that's cool.
And then before you throw it away, look at it again, you might see something interesting. And so, there's several papers throughout my career that are directly stemming from those types of observations, which, I don't know why. I don't know how to train someone to, other than just telling them that that's a thing that you can look for. But yeah, that's in terms of sort of like the serendipity of science, which I think is something you enjoy too. That's just one of those. Yeah, it's just one of those things that you have to look for it. But if you don't know to look for it, you're probably not going to look for it. And so it's one of those things that you can just sort of say, here's an approach to sort of generate those kind of serendipitous observations and then do something with them. And, sometimes interesting stuff comes of that, but that's all I got.
Yeah, no, thank you so much. No, I really hope that folks take notes across the board of all kinds of different little bits of wisdom from this. This is a really cool inaugural episode, and
okay, good.
In many ways, fitting of the series title. Are you familiar with Blade Runner by any chance?
I saw it's Tears in the Rain, or
like Tears in Rain is the final quote.
Tears,
of the movie. Yeah.
Tears in Rain. I guess I'm not. I'm not averse to old science fiction. I just don't know Blade Runner that well.
Yeah, no.
I thought it was a Willie Nelson reference, actually, for Blue Eyes Crying in the Rain, perhaps.
No. Just near the end of the movie, the central antagonist was saying that all of his memories he's about to shut down as a pre-programmed humanoid that has like a 4-year life expectancy, and there was just like an insurgency of these like slave-like creatures that are humanoid that just wanted more life, right?
Ah.
And yeah, and so they were fighting back because there's this like group of folks called Blade Runners that would decommission those humanoids. So there's a big
Mhm.
battle between Harrison Ford's character and Rutger Hauer's character. And at the end, and near the very tail end of his life expectancy, obviously giant spoiler alerts from original Blade Runner, he had this beautiful monologue where he improvised the very last line of that monologue, saying he recanted all of the things he's seen in his life, really things that you can't
Uh-huh.
imagine. And it says all those memories are going to be lost, like tears in rain, right?
Oh, yeah.
And so a lot of the purpose of this series is to catalog these experiences of theses that would otherwise just be put on a shelf, and the
Yep.
people behind it. So I'm really hoping that I'm going to get more folks just like you, and to tell a more personal tale, right? Just so that
Yeah.
folks can relate to. But thank you
Yeah.
so much for this. This was
Yeah,
very,
you
very
bet.
cool.
Absolutely.